Replication. All organisms must duplicate their DNA with extraordinary accuracy before each cell division. DNA replication is the process in which genetic material that is encoded in a DNA sequence is copied so that it can be passed to new cells and offspring through. For humans, DNA replication is the reason we bear similarities to our parents and relatives. While the aim of replication is to yield an identical copy of the original DNA, errors in the process make this impossible and lead to mutations. Although these mutations can be fatal to cells, many are not and act as a means of diversifying cell offspring. The replication process is essential for cell growth and reproduction. The process of DNA replication begins when specialized enzymes pull apart or unzip the DNA double helix. As the two strands separate, the purine and pyrimidine bases on each strand are exposed. The exposed bases then attract their complementary bases. Deoxyribose molecules and phosphate groups are present in the nucleus. The enzyme DNA polymerase joins all the nucleotide components to one another, forming a long strand of nucleotides. Thus, the old strand of DNA directs the synthesis of a new strand of DNA through complementary base pairing. The old strand then unites with the new strand to reform a double helix. This process is called semi-conservative replication because one of the old strands is conserved in the new DNA double helix. DNA polymerase joins nucleotides in a 5-3 direction on the leading strand. However, DNA polymerase does not elongate a DNA strand in a 3-5 direction. Therefore, the 3-5 strand, called the lagging strand, is synthesized in short segments in a 5-3 direction. These short segments placed on the lagging strand are Okazaki fragments and are ultimately joined together by the enzyme DNA ligase to form a new DNA strand. DNA replication occurs during the S phase of the cell cycle. After replication has taken place, the chromosomal material shortens and thickens. The chromatids appear in the prophase of the next mitosis. The process then continues and eventually two daughter cells form, each with the identical amount and kind of DNA as the parent cell. The process of DNA replication thus ensures that the molecular material passes to the daughter cells in equal amounts and types.